When we look at people around us, we find a great deal of variation. For example, if you just turn your head and see all those who are sitting next to you, you find that there is a difference in with respect to hairstyle. Somebody has straight hair, somebody has a curly hair, somebody has little brownish hair, somebody has black hair. There is of course a difference in terms of the slant of the nose, the lips, the eyes, the full facial feature, height, weight, everything. When we look at the characteristics, the psychological traits of an individual, you find a great deal of variation in the behavior of individual. For example, you behave differently when you are in your house compared to when you interact with a particular instructor. With the same instructor, today you behave differently, tomorrow your behavior might change. And in the same day, say in a 3-hour slot, you meet 3 different instructors, you react differently to all 3 of them. This type of a variation that we see in our behavior could either be genetically determined or it could be because of the environmental conditions. Today, we would be talking about the genetic and the environmental basis of human behavior.
गाजर 5 रुपए किलो, 5 रुपए किलो।
When you looked at this video, you found a great deal of variation. People were different in all spheres, irrespective of their look, even their interaction. There was no point where you could say that Mr. X was exactly like Mr. Y. Look at this child. Genetically, when you look at this child, you are primarily interested in looking at how the chromosomal constitution of this very individual has been determined. For example, you look at the color of the child, you look at the different features of the child. Besides looking at the observable characteristics of the child, one could even go to the extent of looking at the genetic makeup of the child. Behaviorally, what we do is not looking at the way the individual looks like, not looking at the genetic code of the individual, but primarily looking at the behavioral manifestation given by this child. From the behavioral science point of view, when we look at a human being, we try to understand his or her personality, intelligence, memory, problem solving ability, thinking style, and so forth. We are more interested in understanding the ability of what this very individual can do, and also the way he can behave. This could either be determined from the cultural viewpoint, or it could be a situational demand. But then behaviorally, what we are more interested in is not only the ability of the individual to perform certain desired tasks, but also to behave in certain desired way. From a purely genetic viewpoint, one would look at these traits and behavioral outcomes and how gene regulates them. For example, in the previous case, I was telling you that what we are interested is more the behavioral outcome, say like the personality of the individual, the intelligence of the individual, the memory of the individual, problem solving ability of the individual, the thinking style of the individual. But genetically, one would primarily be interested looking at these traits and how the gene determine these traits, how these behavioral manifestations are controlled or mediated by different genes. So, all of these would be looked at with reference to the concepts that we would be coming at right now, the nucleotides, when we talk of adenine, thymine, cytosine and guanine, what is popularly called as the ATCG, the bonds that are formed. When we talk of genetic inheritance, primarily what we talk about is the 23 pairs of chromosomes that human beings have, and out of the 23 pairs of chromosomes that human beings have, 22 pairs are autosomes and one pair is the sex chromosome. Sex chromosome basically only plays the role in terms of determining the sex of the individual. So, the Y chromosome coming from the father and the X chromosome are coming from the mother, if these two meet, then fine, you have a baby boy, and if you have the X chromosome coming from the father also, the XX combination that primarily comes gives birth to a baby girl. Sex pair plays only that role, where you have the determination of the sex of the individual, whereas the remaining 22 pair of autosomes, they primarily decide your entire behavior. Gradually, we will explore all of them, and we will take a couple of examples to see how these 22 pairs of autosomes determine several things in one's life. As I told you that we all have 23 pair of chromosomes, 22 pairs are autosomes and one is the sex chromosome. Diagrammatically, if you look at, this is how the chromosome looks like, and when we talk of genes, this is what people would be referring to. You must have seen both these diagrammatic representation of the chromosome and the genes several times. From a behavioral viewpoint, when we talk of behavioral genetics, looking at the genetic basis of human behavior, we do talk of the passage of traits from parents to children. Say the father looked like this and therefore, the child also looks like this. You must have heard this in general conversation, when people proudly claim that, see, my son exactly looks like me, or my daughter is just my replica. This is how you have a great degree of resemblance between the offspring and the parents. This has to do with the chromosomal transmission, the genetic transmission. What is also important is the selective demands that the environment put on you. So, what you have is what your environment demands from you. For example, if you are in a terrain where it is a hill and the productivity level is not very high, what environmentally you are supposed to do would be little different from if you are in the plain region where you have abundance of water, you have very fertile soil, lifestyle would differ, the food intake pattern would differ, the clothing would differ, even the selection of the words, the style of communication, everything would vary. So, human beings basically live at the intersection of the two. You have your genetic predisposition and you have your environmental demands, and therefore, nature and nurture, they act together to shape any individual. Nothing is purely genetically determined and nothing is purely on the hands of the environmental demand. Basically, you have interaction between the two. Let us talk of behavioral genetics in a little more detail. We have heard about the DNA, right now we talked about it. These are the deoxyribonucleic acids, which comprises of 4 different nucleotides. These nucleotides are also called as bases. They are adenine, thymine, guanine and cytosine. And primarily, when one looks at the bonding between these nucleotides, the paired bases of these nucleotides, we look at the alphabetical representation, the AT and the CG bonds. So, these nucleotides, they pair together, adenine pairs with thymine and cytosine pairs with guanine. The genes are actually the segment of DNA that carries instruction for making any specific protein, at times even for RNAs also. And this is how it actually looks like. You have adenine, you have thymine, you have cytosine and you have guanine here. When they combine together, so ATCG is the bonding pattern. So, when adenine and thymine combines together, this is how they look like, and when cytosine, guanine, they combine, this is how they look like. So, when we look at these basic pairs in the DNA sequence, you actually have this. So, every thread that you find here in the this double helical structure of the DNA, the intermediate lines that you find here, you have the ATCG bonding. Now, a gene might have a few hundred to over million bases of pairs, and as we have discussed that human beings have 23 pairs of chromosomes. One chromosome has few hundred to several thousand genes. So, you imagine when we have 23 pairs of chromosomes and we have 100 to few thousand genes, then this means that we have approximately 30 to 40,000 genes and then also approximately 6 billion DNA base pairs, and that is the reason why we have so much of variation among ourselves. When you look at the world around you, when you looked at the video right now of the difference between individuals, you did not find any two individual exactly being a replica of the other, and this primarily means because you have great degree of possibility that we all can vary. Two important constructs needs to be understood over here. We have talked about the passage of genetic traits from the parents to the offspring, but it is not that the full chromosome gets transmitted to the next generation. There is a concept called mutation, and little later we will also come to a concept called recombination. Mutation actually refers to changes in the genomic sequence, and such changes could range from a single DNA base to a segment of a chromosome. These mutations could be of two types, the germline mutation and the somatic mutation. Germline mutation is hereditary mutation, basically it is inherited from the parents. So, right from the beginning you have the germline mutation, you have a mutated gene with you. Somatic mutation are actually acquired mutation. These are the mutation in DNA of certain cells due to environmental factors. For example, you are overexposed to ultraviolet radiation, and this overexposure to ultraviolet radiation could cause acquired mutation, could cause a somatic mutation in you. So, we have two types of mutation, the germline which is inherited and the somatic mutation which is acquired. You can very clearly see that even in mutation you have something that is with you right from the birth, whereas something that has come because of the environmental influence. Although all of us share obvious body parts, all of us have nose, mouth, a pair of eyes and so forth, but none of us are alike. And the variation between human beings are because of the recombination and the mutation. Mutation we have discussed. So, when you look at even at the identical twins, even they might have difference in the shapes of their face, although they share the same DNA. And they also differ in terms of metabolism of specific substances, once again even though they are sharing the same DNA set. When we look at recombination, in normal circumstances one inherits one chromosome from the father and the other chromosome from the mother. Now, this process is repeated in the next generation as well. However, this chromosome is not the intact one inherited from the father and the mother. It is a combination of the two, and therefore, we always will have a recombination, where what you have inherited again gets combined with something else. So, recombination is the genetic mechanism producing more genetic variation. Therefore, even offspring of the same set of parents, say you have a mother and the father, generation 1, generation 2, generation 3, and when you look at over generations how the changes have taken place, because of these mutation and recombination, you do not find similarity between any two member of the same family. So, such variation would not have been possible had the whole chromosome been transmitted. So, if you have the whole chromosome getting transmitted, recombination would not have taken place, and therefore, many people would have been alike. Now, you have one chromosome coming from the father, you have the other chromosome coming from the mother, and then you have a mutation taking place in each of these chromosomes, single threads of the chromosome, and then even in the recombination across generation it gets recombined. Therefore, mutation and recombination creates great degree of variation that we see in our world. Now, variation as such is desirable as the likelihood of survival of the species in the changing environment increases. Let me give you an example. If suddenly you realize that the overall environmental condition have grossly changed in this world, we will all have to compete with the environment, we will have to adapt to the environment, we will have to think of better and new strategies that can help us survive. Now, when you look at the survival strategies, the planning that human beings have to do, the new set of behavior that they exhibit in order to survive, few would succeed, few will not. Now, if there was no variation between individuals, then either all would have survived or all of us would have died. This never happens. Some succeed and some do not. Now, therefore, it is always considered that when you have variation, which is of course a byproduct of mutation and recombination, it is always good, because overall the chances of survival of a species increases manifold, because you have no variable degree of change between the individuals. So, what is not accepted in a given situation could suddenly change, because you have somebody else who has much better strategy that one is adopting in order to survive in the world. And therefore, despite all types of changes that you see in the world, you always have people who survive. If you look at the whole history of the animal kingdom, you realize that few animals have become extinct, whereas a few animals could survive, and all those who survived actually had to undergo great degree of modification in whatever they are, and the way they interact with their immediate environment. So, variation makes important contribution that way. Before we go into the details, now let us look at certain important terminologies. These terminologies are polygenic, phenotypic, genotypic, dominant and recessive. So, before we go into certain other details, we will be I will be telling about these important terminologies, what they imply. Polygenic, as you can make out, that this is a term used for traits whose phenotypic influences are actually derived from more than one gene. So, you have basically a phenotype, right now I will come to it, it is just like say the way you look like, but then all observable things would come under this category, but then it is not that one single gene determines it, but you have a combination, and therefore, each of the gene that combines in a polygenic condition will actually have some additive effect. Say if you have a mutual influence of 4 genes, this very phenotype will be considered as a polygenic outcome, and here all of the 4 will have some contribution to this behavior, to this trait, and therefore, these genes the combination is where all of them are making small, but additive contribution. Phenotype or phenotypic traits are actually the observable traits of the living organism. When you look at individuals, the way they look like, say when you look at me, the way I look like, when you look at your friends, all those who are in and around you, you are actually looking at their phenotypic characters. So, all that is observable, all the traits of the living organism are the phenotype. When we look at the genotype, this represents the genetic makeup of the individual, and therefore, it cannot be observed. Now, you can look at my facial features, you can look at my height, you can look at my weight, you can look at my color, you can look at the texture of my hair, but then what type of genes I carry, whether they are dominant, whether they are recessive, okay, how they have combined, none of this can be observed. So, the genotype is never known, never seen, but then of course, it can be studied through other means, but phenotype is something that is generally observable. Now, we also look at the characters with respect to whether they are dominant or not. Dominant traits are basically those traits where the genes which has been inherited makes an outward display of that trait. So, those genes whose inheritance makes the display of the trait, those are called dominant genes, and of course, they do not allow the expression of the recessive genes. Say for example, color of the eyeball, for example, you must have seen people with black eyes, with brown eyes, with bluish eyes. Now, quite possible that one of the parent had a black eye, the other parent had a brown eye, and then this child has a black eye. Even though the child genotypically could be carrying the brown and the black eye traits with him or her, okay, phenotypically what is visible is the black color of the eyeball, and therefore, the gene determining the black eye actually has become dominant, and it has not allowed the expression of the brownish tint in the eyeball. And of course, because one gene is dominant, definitely it implies that some other genes would be recessive. So, as the name suggests, they are masked by the dominant genes, they are not expressed, only they get expressed in the condition of homozygous states. Now, where you have the same zygotic things, and where these recessive genes get expressed are actually the homozygous state, where you do not have a dominant gene, and in the absence of a gene that can dominate your expression, these recessive genes, although they are recessive, but they get expressed. Let us understand genetic inheritance of traits by this example. Here you have the father and the mother who are carriers of a given disease. The blue color represents unaffected individuals, whereas the black color represents individuals affected by the disease. Combination of both the colors represent that the individuals are carrier of the disease. Here, when both the parents are carriers, the first offspring is completely unaffected by the disease. The second and the third offspring are carriers, whereas the fourth offspring is affected by the disease. Now, the big question is to understand the relative contribution of genetic inheritance and environmental determinants of any human behavior. We have seen that we have the dominant genes, we have the recessive genes, we have seen how a combination of genes they affect the behavior in the polygenic state. There are dominant genes that does not allow the expression of a particular trait, but then you also have selective demands that are put forward by the environmental conditions, and for the healthy survival one has to actually show competence that irrespective of the adversity of the environmental condition, one can survive. So, the big question for anybody in behavioral sciences to understand is the relative contribution that the genetic inheritance makes and the environmental determinants they make. Genes are responsible for transcription and translation of DNA into proteins, which in turn contribute to brain development. Now, in behavioral sciences, we do look at the behavioral outcomes with respect to the anatomy of the brain, with respect to the neurotransmitters, with respect to the receptors, with respect to the neuronal interconnections, and all of them, they all are regulated by the genes. Environment can modify or disrupt actions that are genetically coded. So, what is interesting here, that every time when you look at these slides, somewhere it indicates that even though your brain anatomy is responsible, your level of neurotransmitters is essential, your receptors, the neural interconnections, all of them are important if you are coming forward with a set of reactions, but then the genes are responsible for regulating it. But then even though genes are responsible for regulating all of them, you can have environmental disruptions that can modify or completely disrupt the action that is intended by the genetic code. Let me give you an example. Variant of dopamine receptor gene may be associated with ADHD and thinner brain tissues in areas handling attention. Now, what is interesting is that we have the polymorphism of DRD4, that is the dopamine D4 receptor gene, which is known as 7 repeat form. Now, all those with 7 repeat form of DRD4 have been found to have thinner tissue in right orbitofrontal or inferior prefrontal and posterior parietal cortex. Now, interestingly enough, in behavioral sciences, you come across lot many studies on ADHD, the symptoms, the management of ADHD children. But what is interesting is that although ADHD is now reflected in the form of certain behavior, what is interesting is that attention deficit that ADHD actually deals with has to do with the brain tissues in certain area, and then you have the thinning of that the brain tissue in those areas, and thinning of the cortical areas is actually mediated by the 7 repeat form of DRD4. Here what this set of knowledge informs us is that you have a behavioral manifestation which is a clinically challenging issue, but then it is something that is purely genetically determined. Although you consider that the brain is responsible for such type of ADHD symptoms, but then the attention deficit that is reflected in the ADHD kids actually has to do with the attention areas in the cortex, and then you have the thinning of the tissues, the brain tissues in those areas, which again has a genetic control. So, indirectly one can imply that you see ADHD also has a genetic control. Let us take another example. The amount of gray matter in the frontal cortex is determined by genetic makeup of the parents. Now, this in turn is correlated with one's cognitive ability, because frontal cortex has to do with certain set of cognitive abilities. This means indirectly what I am saying is that the type of cognitive abilities that I display has to do with the genetic makeup of my parents. Again, cognitive abilities can now be linked to genetic predisposition. The brain areas controlling reading skills and language have been found virtually identical in identical twins. Okay. Basically means that they are sharing exactly the same genes, and the brain areas that control the reading and the language skills, okay, they are identical in those identical twins. Now, in siblings, it has been observed that 60% of difference remains there in the case of siblings, whereas in the case of identical twins, it is virtually identical. And this helps us understand why certain disease runs in family. Dopamine and serotonin are involved in muscle control, memory, sleep and emotion behavior. Okay. So, till now we were looking at DRD4 variation, and we were looking at how it affects the brain anatomy. Okay, now we are moving from brain anatomy to neurotransmitters. Now, dopamine and serotonin, these are the two most important neurotransmitters that we have, and they are involved in a series of activities, right from voluntary muscle control to memory to sleep and to all types of emotional regulation. Now, they are also associated with some illnesses, say such as Parkinson's disease, such as mood disorders. Now, TPH2 gene in mouse has been considered to produce 50 to 70% less serotonin. Of course, we do not know the role of TPH2 gene in human beings, but in the mouse it is very interesting. Know that you have 50 to 70% less production of the serotonin, and this is an example of naturally occurring genetic difference controlling production of neurotransmitters. So, you just have the TPH2 gene in the mouse, and then because of the presence of this TPH2 gene, you suddenly realize that the overall serotonin level that is produced drastically reduces between 50 to 70%. And what we had discussed was that serotonin has an important role to play with respect to series of behavior, normal patterns of behavior such as motor control, such as memory, such as sleep and emotion regulation, to the types of mental illnesses such as mood disorder and Parkinson's disease. This means that the whole set of these either the two abnormal behavior that we are talking about here, the illnesses, mental illnesses, and the whole set of normal set of reactions that we are talking about has to do with the TPH2 gene. It is not known whether TPH2 varies in human or whether different variations affect production of serotonin in human beings or not. Let us take another example, the FOXP2 gene. Human beings learn vocalization through imitation. If you have seen the babies, how they grow, how they acquire language skill, you see a small newly born human baby which can just smile but can't create any sound note, and gradually that converts to some accidental sound notes, and those accidental sound notes converting into babbling. Say one accidentally says da, and then the elderly members of the family start saying yes, da, dada, dada, and actually for us dada means grandfather, that is the Hindi language. Now, or say a Bengali will say no, dada is the elder brother. Now, dada has a meaning, but this da that the child spelled was actually an accidental sound which gradually gets encouraged, and in the due process the child starts imitating the language spoken in his or her immediate environment, and this is how one develops complete language ability. Now, development since 2001 has elucidated how a single gene contributes to our oral communication. We have talked about mutation. Now, mutation in the FOXP2 gene has been observed in the KE family that has caused verbal dyspraxia. KE family study is very interesting because couple of members of this very family, they have displayed certain anomaly in terms of normal communication, what is called as the verbal dyspraxia. And once again, what is responsible for verbal dyspraxia in this family, the KE family, is the mutation in the FOXP2 gene. So, this is actually a developmental disorder with a significant disruption of speech and language. I am referring to verbal dyspraxia. Now, the inheritance of FOXP2 is consistent with an autosomal dominant mutation. It leads to the disorder of orofacial movement, thus causing speech and language disorder. So, when you have the orofacial movement of the areas here, the muscles here, this gene, actually this mutated gene, the FOXP2 gene, somewhere inhibits it. Although FOXP2 gene is not the gene for speech and language, but its dominant effect can be seen. Till now, what we have been talking about is that you take a particular gene, and then either its mutation or the presence of the gene that will lead to certain type of anomaly in the development of the brain and or the whole production of the neurotransmitter, and this in turn will affect your behavior. For example, we took the FOXP2 gene condition where the orofacial muscle had some problem in terms of movement, and therefore one exhibits the problem what is called as verbal dyspraxia. We took the other example where you have another gene and then you have 50 to 70% less production of serotonin, or say some other gene where you realize that the attention area of the brain has a thinning of the brain tissues. So, basically what we were actually referring to is that you have a complete genetic control over the behavioral manifestation, whether normal or abnormal. Let us take another example. At birth, human babies are barely developed. The intrauterine conditions are very conducive for the child, whereas the external environment is full of unprotected sensory inputs. So, a growing fetus in the mother's womb is in a very conducive condition, where right from getting food to protection from the external threats to all biological processes are taken care by the mother in the womb, and it is a very conducive life. Once the child comes out, the child experiences the unprotected sensory inputs, and the beauty of this unprotected sensory input, the exposure to it, is that this allows the nervous system to develop in harmony with the environment. Had human babies developed fully in the mother's womb, they would have had experienced difficulties with respect to adaptation. Now, I am endowed with several capabilities, but then I am exposed to an environment where I have n number of unprotected sensory inputs. I have no defense to it, and because I do not have defense against these sensory inputs, therefore, I will start adapting to it. And the moment I start adapting to these external demands, actually what happens that my nervous system gets an opportunity to develop in harmony with the environment. Because my nervous system is developing in harmony with the environment, therefore, the chances of my survival and my healthy survival increases, and because the chances of my healthy survival increases, therefore, me remaining productive in my life, remaining satisfied in my life, coming forward with the desired set of social behavior, those chances increases. The little cognitive development at birth is enriched by life experiences, okay, and this also leads to developmental variation. And we had discussed some time back that once you have the variation, of course, we were talking with respect to genetic variation. Once you have variation, the possibility of your survival in this threatening world maximizes. Here, what we are referring to is that you are born in a condition where you have too many external demands, too much of sensory inputs. Okay, you are full of the unprotected things, but then even though you are not in the conducive setup, even though you are full of these extra exposures to environmental conditions, you provide an opportunity to your nervous system to grow, to develop with respect to what the demand of the external world is. And therefore, the way you cognitively develop, you remember right in the beginning we saw that what we are interested in behavioral sciences is actually looking at the personality makeup, the thinking style, the critical thinking, memory, okay, all these abilities, the demonstrated abilities. Now, all these cognitive abilities that one looks forward will certainly be enriched, because one has an exposure to variety of experiences in life. And of course, this will lead to developmental variation. Once again, the chances of your survival maximizes. Let us take an example here. Genes specify the growth hormone and the receptors for them. They also specify the thyroid hormone and the insulin. Growth of bones is also specified by some other genes. So, when you look at this slide, actually what it tries to say is that you have complete genetic control over all your growth hormones and the receptors for them, and we also have the other genes which will actually determine the growth of your bones. Now, with these genetic predisposition also, one's height might not achieve full genetic potential if he or she is experiencing starvation during puberty. So, although you are endowed with certain genetic makeup wherein your genes has given you the best of the thyroid hormone, has given you the best of the growth hormones and the receptors, but then because of the environmental condition that you are experiencing, because you are experiencing dearth of the nutrition in your life, you are experiencing starvation in your puberty stage, and therefore, despite the fact that you are genetically predisposed to be very tall because of because of the growth of your bones, okay, still you do not achieve the potential height that you could have otherwise achieved. This actually reflects the condition that even though you are genetically predisposed for something, environmental condition can disrupt it. It is difficult to map human behavior to a gene. There are genetic markers of abnormal behavior, but the genes affecting abnormality do not guide normal behavior. Okay. So, remember one thing, you might come across several studies where you have actually the genetic marker for a set of abnormal behavior, which could otherwise mean that this gene probably affects the abnormal behavior, but then one should not construe it the other way around, understanding that if this gene affects abnormal pattern of behavior, this means know that this gene will also have to do with the normal behavior of the individual. So, they do not guide. Let us take an example of phenylketonuria. Phenylketonuria is a metabolic genetic disorder, and here one experiences deficiency in phenylalanine hydroxylase, the PAH, which is actually a hepatic enzyme. Actually, in a normal situation, what is expected is that your phenylalanine will actually metabolize to tyrosine, but this does not happen, and instead of facilitating the metabolism of tyrosine, phenylalanine actually gets converted into phenylpyruvate, or also called as phenylketone, and that is from here the name comes phenylketonuria.
Formation of new cells and repairing of tissues require proteins. Proteins are made up of amino acids. Unwanted amino acids are broken down and eliminated out of the body through urine. Phenylalanine is one of the amino acids. Individuals with phenylketonuria cannot process phenylalanine and it starts building up in their blood. This in turn adversely affects neurons. Children with untreated phenylketonuria tend to have smaller than average head. This is known as microcephaly.
So, when you take phenylketonuria, it is caused by gene mutation, which is also manifested in intellectual handicap and behavioral abnormalities. So, those who have phenylketonuria, you find that they have certain intellectual impairment. You also observe that they have certain behavioral anomalies. Okay. And of course, phenylketonuria is genetically determined. However, the mutated gene causing phenylketonuria is not responsible for normal intelligence or normal behavior. What I am trying to say is that one can develop this fallacy that say if a gene actually makes phenylalanine convert into tyrosine, okay, which does not happen, and therefore, phenylalanine gets converted into phenylpyruvate, okay, and therefore, one experiences behavioral anomalies or intellectual deficiencies. Okay. The fallacy could be that one can reconstruct to say that fine, if this gene affects the intellectual capability, or if this gene affects behavioral anomalies, this means that if you did not have the mutation of this gene, okay, it should be responsible for normal intelligence, but that is not true. And similarly, you cannot trace the absence of mutation of this gene to normal behavior. This cannot be done. Now, you look here, okay, you have the carrier father, you have the carrier mother, okay, and irrespective of that fact, you realize that one of the offspring of that parent gets affected, whereas the other remains unaffected. Most human behaviors are complex and they comprise of a range from normal to pathological. For example, when you feel sad, this is a normal behavior. All of us feel sad in our life, but then one might be sad to the extent that it can be clinically considered as depression. Now, remember, depression is actually a pathological symptom, whereas feeling sad is not so. And therefore, what I am trying to imply is that our behavior is too complex. When you stretch the same behavior and make it go to the extreme, you realize that it loses gradually what is called as the desired or the normal set of behavior. And therefore, what is okay, acceptable, clinically acceptable, okay, need not be, when you stretch it, need not remain normal at all. We have seen this example. All of us have experienced sadness in our life, but then very few become depressed. Depressed is, now I am referring to clinical depression here. Francis Galton was the man who came forward with a concept called eugenics, that is the well-born. And in the early 20th century, he focused on the genetic causes for mental retardation, alcoholism, criminal behavior and immoral behavior. So, primarily what Galton was trying to focus was that for all of these aberrations that you see in human beings, you have a gene that is responsible for it. So, if you are genetically well born, you will not display these anomalies, you will not display these set of what you call as the uncelebrated behavior. You won't show mental retardation, you won't be alcoholic, you won't become a criminal, and you would completely comply to the social norm, and you won't come forward with immoral set of behavior. When we meet next, we will be talking with a special reference to alcoholism, with a special reference to criminal behavior, and we will be primarily looking at trying to testify, is this whole concept of genetically well born correct? Is it that alcoholism or say criminal behavior is genetically predisposed, or is it that we pick up from our environment? This is now time for us to recapitulate whatever we have discussed about the genetic predispositions and environmental determinants of human behavior.